My presentation is about potential and current applications of genotyping at MPG using non-invasive genetic sampling to genetically tag organisms. Microsatellites are short tandem repeats or simple sequence repeats. They're usually between 2 and 6 base pairs of DNA. Dinucleotide repeats are the most common, just shown here. They're 2 base pair repeats repeated over and over again, and usually that region is between 100 and 200 base pairs in length. So we like microsatellites because they are some of the most variable regions within the genome. They have a high rate of polymorphism within a given population. That is, the repeats can have many different lengths. This hypervariability allows us to differentiate at an individual scale. And then just to be clear, the variability between individuals is in the length of the repeat. With microsatellites, the mutation rate is equal between males and females, which means we can use the same region for both sexes. And the mutations are heritable. They follow Mendelian inheritance, which means we can look at parentage. So before we move forward, I just want to go over a couple of definitions. Loci are specific regions of DNA on a chromosome that contain the microsatellite or region of interest. Loci can have many different forms. Each form is called an allele. For each locus, there are a number of possible alleles within a population. This usually depends on population size and genetic variability. Homozygous individuals are organisms that have 2 copies of the same allele at a single locus, and a heterozygous individual has 2 different alleles at that specific locus. So for example, say we're looking at a microsatellite output for 4 unique individuals, 1, 2, 3, and 4. The region of interest in this case is locus G10b, which has a range from 96 to 116 base pairs, which is on the horizontal axis. And at each locus, an individual can have up to 2 forms corresponding to the 2 parent chromosomes. These forms are called alleles, and they're represented by the peaks shown here. The horizontal position of the peak, again, represents the length of the allele, and individual 1, you can see, has 2 alleles at 102 base pairs and 110 base pairs, making it heterozygous for locus G10b. And then individual number 4, on the other hand, is homozygous with 2 copies at locus 110. So just to give you a very general picture of the process of microsatellite genotyping from start to finish, first, you begin with genomic DNA from your target population. You use the genomic DNA to find a good simple sequence repeat, one that's easy to amplify, preferably not too long, and that has good sites on either end for primer development. You then choose your loci that will tell you most about your population of interest. You want to make sure that you have enough loci to represent all the variability within the population. You want your loci to be species specific most of the time, so you're not amplifying non-target animals. And you usually want at least 1 loci that's sex specific, so that you can differentiate between males and females within your population. Then you can develop your primers and assign fluorescent labels. After that, you're ready to begin processing your sample, first by isolating DNA. Next, you use PCR to amplify your chosen loci. Then fragment analysis is used to identify the loci within your sample, and it gives readings according to band intensity and outputs an electropherogram. And then you look at these electropherograms for each loci that you amplified, and you can begin to determine the fragment lengths and the differences in your samples. And so then you can combine all your microsatellite data for each individual, and this becomes its genetic fingerprint. And then you can run each multilocus genotype through various error checking softwares. So from there, we can start asking questions about our population of interest. We can begin first by identifying the individuals within our population through hair samples, through scat, or sometimes tissue. We can look at parentage. We can look at the degree of relatedness between individuals. We can look at the demographics and the genetic structure of our populations. We can assess the effective population size. We can look at gene flow between populations, or the effects of land management or other environmental factors on the genetic structure over time. And of course, just learning about animal behavior and movement. So there are numerous examples in the literature. One, looking at parentage, did a study on the secret sex lives of sage grouse using microsatellite analyses to reveal that a single male grouse can father multiple broods within a single season, and that a single clutch will have multiple fathers up to 10% of the time. Another study looking at environmental influences used data from 10 microsatellites of almost 900 house sparrows to show how food availability influenced the genetic structure. And I think with the extensive phenology data that we have at the ranch, there's a lot of potential for studies similar to this. For example, is there higher genetic variation in small mammals in areas with earlier flower or seed development on the ranch? And then looking at gene flow, another study by Sawaya et al. Used microsatellites and multilocus genotypes to show gene flow over wildlife crossing structures in Banff National Park. And so one other nice thing about microsatellites is that microsatellite analysis is PCR-based, which means it works with very small amounts of DNA that we usually find in hair samples or scat, molted feathers, or old bones. It works on very highly degraded DNA, so samples that have been exposed to the sun for long periods of time, or weather, or are degraded from enzymes. And also, these samples, they don't have to be collected directly from the animal, which means that there's little to no impact on an organism's natural environment. So which brings us to black bears at MPG Ranch. We started using microsatellite analyses and non-invasive genetic sampling to monitor black bear populations at MPG Ranch in 2012. And then in 2013, we started processing all of our samples in-house. This data will supplement and confirm video and image data that we have already collected. And it'll be especially useful from scat samples where we don't always get video footage of the culprit. It'll allow us to examine parentage and relations. We can start looking at things like mate choice, why a female chooses a specific male bear to mate with. We can look at animal behavior, and then of course demographics and the population changes over time. So this summer we monitored around 12 installed bear traps, or bear hair traps or posts, and around 10 established bear rubs. We collected hair samples from May through October, and the samples were stored in the lab in individual coin envelopes for further processing. And here's just a map of all the hair rubs and hair traps that we have currently on the ranch. So each sample is opened up under a microscope, and we choose hairs based on the hair type and whether or not there are follicles, because the follicle is actually what contains the DNA. The hair itself doesn't have DNA, or it's too degraded. Each sample must contain at least one guard hair follicle or greater than 10 underfur hairs to be considered for further processing. And then all of our adequate samples are transferred to 1.5 milliliter Eppendorf tubes for DNA extraction. And then we take pictures of all of our tube transfers so we can go back and double check the sample order later on. So this is a pretty good sample, what I usually get. Just a big clump of hair, there's pieces of wood, grass, or caked mud. All of that has to first be removed. Otherwise it can inhibit PCR later on. So I go through and I look for guard hair follicles and cut each follicle from the rest of the hair to remove the excess material that could inhibit PCR. And if there's a large clump of soft underfur hairs, I'll usually include that too, because it will often contain large amounts of DNA that you can't always see because the follicles are so small. A lot of times, a sample I get will just contain like 1 or 2 hairs and maybe 1 follicle, and we'll go ahead and process it anyway, just in hopes that we'll get DNA. And then sometimes, very rarely, I'll get a sample like this that has tissue. Unfortunately, though, this came from the female bear that was shot in the Bitterroot that Alan talked about earlier. So for an initial analysis, I chose 6 loci based on previous studies of similar populations, similar species, and nearby geographic locations, and 1 sexing loci to determine whether the individual is male or female. Most of these loci are dinucleotide. And so then we optimize PCR to perform multiple reactions at once, and this is called multiplex PCR. And we use this technique to amplify 7 different loci in a single sample using 14 different primers. Fewer reactions reduces the time spent preparing samples and, of course, the cost of reagents. And then we use fluorescent dyes attached to the primers to differentiate between the different loci within each sample. After PCR amplification, we perform fragment analysis, and this separates the PCR amplicons by size through capillary electrophoreses. And the fragments are then passed through a laser beam, which causes the dyes to fluoresce. The dye signals are then separated by a diffraction system, and a standard curve is used to determine the relative size of each labeled fragment within your sample. So after fragment analysis, this is what your output typically looks like. It's a bunch of peaks of different colors that you have to interpret. The loci often overlap, so that's why you can see why you use the different colored dyes to differentiate between them. Otherwise, it would be a big confusing mess, as I'm sure you can see here. If you look, the longer the fragment length, which is on the x-axis, the shorter the peaks get, and that's a big indicator of a degraded sample, of degraded DNA within your sample. So with such high variation in the amounts of DNA, it can be difficult to optimize PCR, especially when we're processing so many samples, and especially when the samples are old and degraded from sunlight exposure, weather, and different enzymes. For example, there's a very clear correlation between, in SCAT between age since deposit and amplification success. 3 of the biggest problems that we encounter are false alleles, which can be artifacts. Sometimes they're from slippage PCR or they're from contamination that produce a peak where there shouldn't be a peak. You can also have allelic dropout where one allele of a heterozygous individual is just not detected. And you can have mixed samples, are samples containing DNA from more than one animal. And in our case, this can happen on the bear rub itself, having multiple bears rubbing on the same rub. So this is what a really good peak looks like from a single allele. You get your true peak, which is the tallest, and then you get a stutter, which is typical in all samples, no matter what the quality. In dinucleotide repeats, though, the stutter is very easy to identify because it's usually 2 or 4 base pairs less than the true peak. But sometimes you will get samples where the 2 alleles overlap and form a composite stutter, and that can make it pretty difficult to interpret. So here's an electropherogram of locus G10B from 3 separate amplifications from the same individual. The true peaks are indicated by the arrows, and you can see the individual shows up as heterozygous at this allele in the 2 bottom amplifications, but then homozygous in the first amplification. In cases like these, it's very easy to mistake one individual for 2, and this can lead to severe overestimation of your population size. So in order to reduce these types of error within our samples, we used a multiple tubes approach. Because of the high potential for error, reproducibility in the situation it can be very important. And so we distribute our extract DNA among several different tubes before amplification, and then we amplify each hair sample at least 8 times to reduce the error. And this makes it so that the probability of observing the same false allele multiple times is less than 1%, excluding, of course, contamination. And it produces a much more reliable genotype when you have this reproducibility. So here's an example from our study. This is bear 966, and it's 4 separate amplifications of the same loci showing the exact same thing, which is really nice to see. Clearly, this is a heterozygous individual at locus G10P with alleles at 163 and 167, and then the stutter peaks that are each 2 base pairs lower than the true peaks at 165 and 161 base pairs. Using these methods, we ended up with 11 total bears based on data collected from just the rub trees alone so far. The 11 different genotypes we found are outlined in blue. And then the error rate that we found per multilocus genotype is outlined in red. And this was less than 5% for most of our samples, which is pretty good considering the small amount of DNA that we're working with. And some of the genotypes actually had 0 error. Right, and our average allelic dropout rate was 3.4%, and the false allele rate was 2.1%, which is comparable to for studies using similar methods. So here's an example of what an electropherogram looks like for 4 different individual bears on the ranch. You can see how it gives a clear fingerprint of each individual, at least this particular locus does. Bear 966 at the top is heterozygous at 167 and 173 base pairs. And then you can see bear 976 has 2 alleles at 163 and 167. Bear 870 is a female, it has 2 loci at 173 and 177. And bear 863 is also heterozygous at alleles 167 and 165. So in 2012, we collected 35 hair samples, 13 of which produced usable genotypes. We obtained genotypes from hair samples at 7 different rubs and 5 different hair traps, although we collected from a lot more than that. And the estimated minimum population that we got for that year was 6 males and 1 female. This past year, our sampling effort was more robust. We had around 12 hair traps and rub posts and about 10 established rub trees. The data we have so far is from the 10 rub trees only, and so far we've processed 159 total samples, and 58 of those have yielded reliable genotypes. And then we still have over 60 samples from scat and hair traps that have yet to be processed. So far, with this data, we can start to look at bear movement and behavior on the ranch. The bear rubs appeared slightly biased toward males, with 4 females and 7 males genotyped total. Bear 966 was a male and was genotyped from bear rub F, indicated by the star, in August. And then in September, it showed up on bear rub H. And then in October, it moved around a lot more, and we saw it at 3 different bear rubs, all near Davis Creek. So this is the bear that corresponds to that genotype, according to Allen's bear rub footage. Here it is rubbing on a tree in August. And this bear is also known as fall black and tan. Bear 863 was also a male, and it showed up at Baldy Draw in April, and then again in Whaley Draw in April. Then we found its genotype in Lower Tongue Creek in June, and then we didn't see it again until October when it showed up in Upper Tongue Creek. Here's a bear that corresponds to this genotype, more famously known as backfloat bear. So, let's see here. Let's go with this one. So we now have a better understanding of which samples are more likely to produce good quality genotypes, so we can eliminate samples with low probability of success and make the processing time much more efficient. We also know now that more regular hair collections are necessary to avoid mixed samples and sample degradation. And with the data that we have, we can start making minimum population estimates and confirming camera data. We're beginning to optimize more primers right now. And once we have finished this initial screening, we can begin to amplify all of our successful samples at additional loci, so we can start asking bigger questions like parentage, relationship between individuals. We can look at mate choice. We can look at behavior in males, maybe versus females. And then, of course, just to continue to monitor the demographics and the population changes over time. And then a big thanks to everyone else who's been a part of this project, Cinepah Wildlife Research Associates, MPG North, MPG Ranch, of course, Bo Larkin and Heather Baltus as well. Thank you.